In this video, I will explain about glycolysis. So glycolysis means means sweet or sugar. It is a Greek word. It means sweet or sugar. Lysis means breakdown. So in this video, how the sugar means sweet, lysis means breakdown. How the sugar will break down and release the energy. So this video I will clearly explain about glycolysis. So the glycolysis is the major metabolic pathway of glucose. So it is nothing. It is a major glycolytic metabolic pathway of glucose. So this pathway is catabolic pathway. What is mean by catabolic means? It means breakdown of macromolecules into micromolecules with the release of energy. So in this video, I explain about how the glycolysis takes place in our cell and how it releases energy and how much amount of energy will be released during glycolysis. So there are two phases in glycolysis. The first phase is preparatory phase, another one is payoff phase. What is mean by preparatory phase and what kind of reaction takes place in this phase? So what preparatory means, prepare to something. So in this phase the glucose is prepared to broken down. So this phase is called as preparatory phase. Another phase is called payoff phase. Payoff phase means, so in case of preparatory phase, some amount of energy is required to broken down the glucose. So in that amount of energy is recovered during payoff phase. So that's why it is called as payoff phase. So there are in this glycolysis pathway, there are sequential reactions of enzymes takes place. So this glycolysis takes place in our cytosol of our cell. So in this pathway, there are 10 enzymes are involved in this reactions. So the enzymes which converts glucose into pyruvate, it is the final product of glycolysis. Then this pyruvate, it can enter into the TCA cycle. Then find that during TCA cycle, the glucose is completely oxidized and release the amount of energy. So it is this glycolysis is also called as partial oxidation of glucose. So in this glycolysis pathway, it will take both aerobically as well as anaerobically. Aerobically means it takes place only in the presence of oxygen. Anaerobically means it takes place only in the absence of oxygen. So different kind of end product will be formed during aerobic as well as anaerobic. In case of aerobic, it releases pyruvate. In case of anaerobic, it produces lactate. So lactate is the byproduct or end product of the glycolysis in the condition of anaerobic. So the first reaction is glucose is converted into glucose 6-phosphate. So here glucose is converted into glucose 6-phosphate. So this reaction is called as phosphorylation reaction. What does mean by phosphorylation? Means addition of phosphate group. So the enzyme is responsible for this reaction is hexokinase. The enzyme kinase is responsible for transferring phosphate. So here the phosphate is transferred from ATP to glucose. So that's why the glucose is converted into glucose 6-phosphate. In case of 6th carbon, the phosphate is added into the glucose. So the phosphate is donated by ATP. So here one molecule of ATP is release phosphate. By the way, it is converted into ADP. So this is the first reaction. So first reaction is phosphorylation reaction. Remind kinase, the word kinase, the enzyme kinase is responsible for only transferring phosphate group. So wherever kinase comes, there is a phosphorylation reaction takes place. So it is easy to remember that kinase means it is responsible for phosphorylation reaction. Somewhat it transfers phosphate from one molecule to another molecule. So the first reaction is phosphorylation reaction. The first 5 steps comes under preparatory phase. The another 5 steps comes under payoff phase. So there is a sequence, 10 sequential enzyme reaction takes place here. So the first 5 reaction comes under preparatory phase, the another 5 reaction comes under payoff phase. So the first reaction is phosphorylation reaction. So the second reaction is the substrate is glucose 6-phosphate. So this glucose 6-phosphate is converted into fructose 6-phosphate. So fructose is the isomer of glucose. Both are the 6-carbon sugars. Glucose is aldose sugar and fructose is a ketone sugar. So isomer is nothing but they are having both same molecular formula and different structural formula. So that's why this reaction is called as isomerization reaction. So remind that what kind of reaction takes place in that step. So the reaction, the enzyme name is somewhat similar to that reaction. So in case of isomerization reaction means the enzyme is isomerized. In case of phosphorylation reaction means the name of the enzyme is kinase. In case of oxidation reaction means the name of the enzyme will be oxidase. So likewise here the enzyme is glucose 6-phosphate isomerase or hexophosphate isomerase. It is also called as hexophosphate isomerase. So here the isomerization reaction takes place. So isomerization means the glucose 6-phosphate is converted into fructose 6-phosphate. There is no addition of any other molecules. Only the isomerization reaction will be takes place here. So the glucose is structurally modified into form a fructose. So it is isomer. So the fructose is isomer of glucose. So it is a second step of reaction. And the third step is the fructose 6-phosphate is converted into fructose 1,6-phosphate. So here there is 2 phosphate molecule in fructose which is in the first position and the another one is 6th position. So here we require 1 phosphate molecule. So obviously phosphate is donated by ATP. Here also 1 ATP molecule will broken down and release 1 molecule of ATP. So I always told that kinase, the enzyme kinase is responsible for that. Phosphofructose, the enzyme name is phosphofructokinase. So fructose phosphate is a substrate. So the enzyme name is starts with phosphofructose. So kinase means that is a transfer of phosphate group. That means phosphorylation reaction. So here the phosphate group is transferred from ATP to fructose. By the way, it is converted into fructose 1,6 biphosphate. It is also called as diphosphate. So this is the third step. The another one is the next reaction is aldolase. Here the fructose 6-phosphate, the 6 carbon sugar is broken down into 2 3 carbon sugars. One is dihydroxyacetone phosphate, the another one is glyceraldehyde 3-phosphate. So it is 3 carbon sugars. So here the 6 carbon sugar, this sequence of reaction, the previous reaction, the 6 carbon sugar is converted into 2 3 carbon sugars. So somewhat the breakdown of molecules takes place. So that is why this pathway is called as the catabolic pathway. So here the fructose 1,6 biphosphate is converted into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So the enzyme which is responsible for this reaction is aldolase. So next step is the glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Both are isomers. Both are the 3 carbon sugars. Here also glyceraldehyde 3-phosphate is aldose sugar and the dihydroxyacetone phosphate is a ketone sugar. Remind that glyceraldehyde. So remember that aldehyde, aldose sugar. Ketone, acetone, it is similar to ketone. So that is a ketone sugar. So both of them are 3 carbon sugar. They are having same molecular formula but different structural formula. That's why it is called as isomers. So the enzyme which is responsible for this conversion dihydroxyacetone phosphate into glyceraldehyde 3-phosphate is isomerase. So these 5 reaction which comes under preparatory phase. So here the glucose is now prepared to broken down to produce energy and the byproduct pyruvate. So the first 5 reactions comes under preparatory phase. So the next 5 reactions comes under payoff phase. Here the ATP is recovered. The first step is the glyceraldehyde 3-phosphate is converted into triphosphoglycerate. 1,3 biphosphoglycerate. So the next step is glyceraldehyde 3-phosphate is converted into 1,3 diphosphoglycerate. So the enzyme responsible for this reaction is triphosphoglycerate dehydrogenase. Dehydrogenase means D means removal, hydrogenase. The removal of hydrogen takes place. So the removal of hydrogen takes place from the removal of hydrogen is from the glyceraldehyde 3-phosphate. So the hydrogen molecule will be removed from glyceraldehyde 3-phosphate. By the way, the NAD molecule will get the hydrogen molecule. By the way, it is converted into NADH2. So NADH2, one NADH2 is equal to 3 ATP. So in this step, indirectly 3 ATP molecules will be formed. So here, like first recovery of the ATP. So that's why it is called as pair phase. So in this phase, in this reaction, one molecule of NADH2 will be produced. So the next step is 1,3-diphosphoglycerate is converted into 3-phosphoglycerate with the help of the enzyme kinase. I already told that wherever the kinase, the name of the enzyme kinase will comes, there is always a reaction of phosphorylation. However, the molecule, the phosphate molecule is transferred from one molecule to another molecule. Here, the phosphate molecule is transferred from diphosphoglycerate to ADP. The ADP will capture that phosphate. By the way, it is converted into ATP. So, one phosphate molecules will be released from diphosphoglycerate. By the way, it is converted into 3-phosphoglycerate. So that's why the phosphate molecule will be removed from first position of the carbon. So by the way, it is converted into 3-phosphoglycerate. The next step is this 3-phosphoglycerate is converted into 2-phosphoglycerate. So there is no addition or deletion of any other molecules. Here also the position of the phosphate will be vary. The position of the phosphate will be vary. So here the phosphate is in the third position and the 2-phosphoglycerate. So these all the reactions are reversible reaction. What is maybe reversible reaction? It may be converted into again the substrate. For example, this 2-phosphoglycerate can able to convert it into 3-phosphoglycerate. So the next step is this 3-phosphoglycerate is converted into phosphoenol pyruvate with the release of water molecule. The water molecule will be released from 3-phosphoglycerate. The enzyme which is responsible for this reaction is enolase. So the final step is phosphoenol pyruvate is converted into the final product pyruvate. Here also the phosphate molecule will be transferred from phosphoenol pyruvate to ADP. The name of the enzyme is kinase. So pyruvate kinase. Here the phosphoenol pyruvate will donate the phosphate molecule to the ADP. The ADP get phosphate molecule from the phosphoenol pyruvate. By the way, it is converted into ATP. So here the phosphoenol pyruvate is converted into pyruvate. That's why the name of the enzyme is pyruvate kinase. So here one ATP molecule will be synthesized. So in this 5 step, there are 2 substrate level phosphorylation takes place. The ATP were produced from the substrate. That means the phosphate molecule will be released from the substrate. From that substrate, the ADP will accept that phosphate. By the way, it is converted into ATP. So that's why this process is called as substrate level phosphorylation. So here there are sequential reaction of 5 enzymes. In case of payoff preparatory phase, the another 5 enzymes is involved in the payoff phase. So now I explain about the energetics. So energetics from how much amount of energy will be produced from glycolysis. Here in the first step and another third step, we require 2 ATP to break down the glucose. So in case of payoff phase, there are totally 5 molecules of ATP will be produced. In first step, 1 NADH2. 1 NADH2 is equal to 3 ATP, and the next step 1 ATP, totally 4. In the final step, produce 1 ATP, so totally 5 molecules of ATP will be produced from 1 molecule of glyceraldehyde 3-phosphate. We are having dihydroxyacetone phosphate always. So this dihydroxyacetone phosphate is converted into another glyceraldehyde 3-phosphate. Again, this sequential of 5 reactions will be takes place. After that, 5 molecules of ATP will be produced. So there are totally 10 molecules of ATP will be produced during glycolysis. So in this 10 molecule, 2 molecules of ATP will be already utilized by the preparatory phase. So that's why totally 8 molecules of ATP will be produced. So this is the elaborate or brief explain about glycolysis. Thank you.